Right, so I'd like to start this presentation with a question I'd like to ask. It's a rhetorical question for me, but the question is, what, this is supposed to work. Yes. What do a topologist and a neuroscientist have to talk about? And of course, I mean, you have neuroscience like many other biological sciences that they produce a huge amounts of data. So you can use TDA, and that can be a very useful tool to analyze that data. In this project, what I want to tell you about is not quite that, but rather we would like, what we are after is trying to produce our own data and then using topological methods for analyzing it. So when I say producing our own data, we are going to produce data inspired by topological methods rather than just biological data, and then using other methods to analyze it. Right, so let me give you a little crash course kind of on neuroscience. So every organism that has a brain has one that is made out of neurons. And neurons look, well, this is a particular type of neuron in the cortex, and they look more or less like this, right? So it's a big, it's a very small thing, but it's very expansive in terms of you know, how many arms it has and how many connections it can make. Neurons come in many types. They come in many, they're different in various parts of the brain. What you see here is a picture of the cortex. And on the cortex, you have 55 distinct morphological types. I'm going to tell you what morphological type means in a second. They are spread across 6 layers. Now the cortex is about 2 millimeters thick. In a rat, which is our main object of study, it's about 2 millimeters thick. And in those 2 millimeters, you have these 55 distinct morphological types of neurons spread across 6 layers. So these 2 millimeters have 6 layers of cortex in them. So here's a picture of the 55 morphological types. As you can see here, these are the excitatory neurons, the ones which react to excitation and spread the news when they get any. And you have lots and lots of inhibitory neurons, these blue ones. You have the layers are here, so you can see that some of the neurons have a certain type, which is the type N, the layer in which it lives. You have the upper layer, the top layer, is mostly inhibitory, or only inhibitory neurons. And then you have the layers 2, 3, 4, 5, and 6. Now, each one of these little objects, which are tiny, is capable of forming anything between a few hundred connections and a few thousand connections. So you get something which is, you get a system, once you look at the brain, you get a system which is really difficult to understand. Because even if you take a small number of neurons, you don't understand how many connections there are between them and how they are connected, is very difficult. And indeed, neuroscience does not know how to do that, except in very few cases. So one case like that is the case of, oops, this is backwards. So in the case of the worm, known as I can't pronounce it, c-elegans or c-elegans, much easier. This worm is 0.5 millimeter in real size. This is an electron microscope image of it. And this is an example where scientists were able to actually identify the entire connectivity matrix for this worm. So it has about 279 neurons in its brain, and about 8,000 synaptic connections among them. And in here, what you see is the diagram of about 4,500 of them. Okay, but in general, you're talking about a brain that has any mammalian brain, for instance, would have millions, tens of millions, sometimes billions, depending on the mammal. Like we humans have between 80 billion and 100 billion neurons in our brain. So doing something like this is practically impossible. For a variety of reasons. Money is one of them, but time is also another one. So that's where the Blue Brain project comes into the picture. So the Blue Brain project does something very interesting. They actually take biological data, they take all this information about the neurons, what they look like, the morphological types. I'm going to make this play again. Oops, I didn't mean that. And they put them together according to stochastic rules, but they're constrained. They're constrained by biological data, by things we know about biology. Okay. They also build a system which actually is capable of being activated. So what you saw there is basically I'm going to play it once more and then explain. What you saw there is a bit of the cortex, which, which you see, if you're close enough, you can see these little spikes. These are the neurons spiking randomly, and then a stimulus comes in, and this sort of storm that you see there is basically the reaction of the system to that stimulus, and then it goes down again. Okay, so what do we have? We have access to the Blue Brain project Reconstruction. So we have that it's built, as I said, a biologically accurate digital reconstruction of the neocortical column. That is, I'll explain what it is in a minute, of a 14 days old rat. Okay, and now each one of these columns consists of about 30,000 neurons with about 8 million connections. Now, what do you do with this? I mean, one thing that you can do with a computer and you cannot do with a real brain is you can actually ask the computer any question you like. For instance, you can extract the full adjacency matrix for that bit of brain, including the strength of connection, the type of the neuron, spatial position of it, of each individual neuron, the spatial position of every individual synapse. You can also extract from this. Full spiking data for each neuron under varying electrical and chemical conditions, and so on and so forth. So with this, you are able to construct a matrix, a connectivity matrix. Sometimes it's stationary. If you want just to look at the structure, it's stationary. If you want to look at more than the structure, if you want to look at the function as well, then you can stimulate the system and get the reaction, get a time series of these connectivity matrices in a way that I'm going to explain in a few minutes. And now once you have a graph, or a series of graphs, you have topological objects. So let me try to explain what sort of topological objects we're looking at. So one typical topological object you get from a graph is, so here's an example for a graph. You get a flag complex. The flag complex is a classical object in combinatorial topology, and it is the maximal abstract simplicial complex containing the graph as its one skeleton. So if this is the graph, then the flag complex of the graph is going to look like this. So the two simplices are the orangey ones, and the three simplices are the purple ones, and all the rest you see is not filled because there's no triangle, there's no three simplex or anything else of the sort. So these remain empty, okay? Which means all you have there really is just the one simplices here. Okay, so that's the flag complex. But we want a little bit more than that, because neurons, the way they're connected, they're not just connections between cells. They are connections which have a direction. So each neuron will have its dendrites and it will have its axons. The dendrites receive data, the axons emit the data. And so there's a well-defined directionality in the way that neurons work, and therefore we want to work with directed graph. So let me review a few concepts here. So first of all, an abstract ordered simplicial complex, which is kind of a variation of just a simplicial complex, is a collection of finite ordered sets, such that if sigma is in S, then for every subset, ordered subset of sigma, tau is also in S. That's all. So it's a straightforward concept. Except, of course, if you have two ordered sets which have the same vertices in them, but they're not in the same order, they're considered by us to be different. Okay? So they're not the same set as it would be in the case of a normal abstract simplicial complex.
So you don't require an order in the whole set of vertices?
No, just a collection of ordered sets closed under subsets. That's it. Under ordered subsets. That's all. Okay? And then, so this is ideal for working with directed graphs. So directed graphs, all I mean by directed graph is a pair, V is the vertices, E is the edges, which are contained in the product. The isomorphic subset of the product equals V. So you have a beginning and an end vertex for each edge. But if you have an edge that has x as its beginning and y as its end, it's not the same as the edge that starts in y and ends in x, and both of them can exist in the same graph. Okay? And then the directed flag complex, by analogy to the ordinary flag complex of a digraph, is the abstract ordered simplicial complex, whose n simplices are just the ordered n+1 tuples of vertices. Okay, so basically, best explained with an example. If you have a directed graph like this, right? So as you can see, you have here triangles, right? You have triangles, and you also have these things which look like three simplices. It's basically the same graph as I had before, except I added now some directionality. It's exactly the same graph, but I added some directionality. And what you get is this. Now, notice the difference. The difference is that here, for instance, you have a triangle. In the flag complex, if I forget directionality, this will be filled in. Here also, I have, in the undirected flag complex, I have a three simplex. Here I don't have it because I don't have the proper directionality. Okay? And similarly here. So it's more or less the same, but not quite. Right? Because you have to take into account whether or not the simplex is directed. Another issue is the issue of having reciprocal connections. So if you look at these three graphs, if you looked at the associated undirected, unordered flag complex, then you get exactly the same thing, just two triangles, because these two edges will basically be the same edge. In a graph, the way we are thinking about it anyway, we're only thinking whether two neurons are connected to each other, not whether they're connected with two edges, because that, I mean, we could do that if you have, for instance, like more than one synapse, and you normally would have more than one synapse. But we don't do that. We only ask the question, are two neurons connected to each other? So this graph, these three graphs will be exactly the same. They have no directionality, and we'll have only one edge here. And so when you take the flag complex, you just get two triangles attached to each other. But here, when you think about it, in this way, then what you get is the following. I mean, this guy here will have a circle in here. I mean, all of them will have a circle as the thing in the middle. But this will realize a two-sphere. This guy here, I'm talking about homotopy type here, not about geometry. As a homotopy type, this is not difficult to see that is going to be a circle. And this guy here is going to be just like a sphere with a hole in it. So what you're going to get here is basically of the homotopy type with point. Right? So the directed flag complex actually does give you a way of saying something about the directionality of the graph that created it. Okay? Okay, so let's go back now to the Blue Brain project, and what you see here is our first surprise when we started thinking about this a few years back. So it was very surprising both to us and to the neuroscientists that we were able to find higher dimensional simplices, higher dimensional directed simplices in the reconstruction of the brain. So what you see here is basically 6 and 7 dimensional simplices. I mean, basically, if you took the realization, it would be 6 and 7 dimensional simplices within the reconstructed tissue. Okay? Now, why is this surprising? Well, it's surprising to the neuroscientists because they never knew that these simplices actually existed. These sort of connections actually existed. As far as they were concerned, 2, 3, 4 neurons connected all to all was very good. Right? 8 of them? Unthinkable. So they were really surprised. And what was surprising to us is that they never actually asked the question. Namely, they had this system, but they never imagined that they could actually look, and it's easy to look, because they have this, I mean, it's a reconstruction, it's a digital system. But they never thought about it, because they never imagined that it could happen. So when we came into the picture, we do this calculation, we built this directed flag complex, and we found these simplices. Okay, so then we started working. We have 42 reconstructed microcircuits based on the data from 5 different rats. Each rat contributed its data and its life to the project. And the data was fed into the algorithm, which, as I said, is partly stochastic. And each rat generated 7 different columns. And then they took the data of the 5 rats, they averaged it, and they put it in again into the system and got an average rat. Okay, and so we have 7 samples from each instance to reconstruct the microcircuits. We wanted to do this analysis of the the simplices and what else with control. So we created as well some Algebraic random connectivity matrices with the same average, the same size and average connectivity as the reconstruction. So the size means about 31,000 neurons, and the average connectivity is 0.8%. Then we did 2 sets of control randomizations. So what we wanted to do is basically to look at something which is closer to the biology, because you can be criticized very strongly for comparing something which is supposed to be as a brain to something which is totally random. Because even you don't have to be religious to realize that something that does amazing things like a brain, even of a rat, is not random. Right? So comparing it to a random is not really fair. So these 2 other sets of control randomizations were something more closely related to the biology. What we did is we looked at the average probability of connection, distance dependent, between the different morphological types. So this data is actually known from the system, and also across different layers, the 16 different layers. So we got 2 different randomizations which are closer to the biology. They have a lot of biological data in them. What they don't have is the actual morphology. Only the probability. So these are probabilistic models, whereas the actual thing is not probabilistic. It actually takes into account the actual morphology, the actual geometry of each neuron, of each individual neuron. And finally, we did a set of randomizations, which which is something which is done very frequently by neuroscientists. We use what is called Peter's rule. Peter's rule is basically connect 2 neurons if their summas are at most 3 microns apart. If they're at most 3 microns apart, you connect them. And then you prune out to get to the correct average probability of connection. Okay? So this is something that most neuroscientists would consider a golden rule. This is more or less the way things are connected. Okay? Right, and then we computed the directed flood complex of the resulting graph. Yes?
So when you're talking about these high-dimensional simplices, is the way we should think of them, there's some kind of path where the neurons are connected end to end, and then also there are connections between ones that are farther apart in this path?
No, let me go back to the picture. It's here. So if you're looking here, for instance, yeah, this guy here is a neuron, one of the neurons here. I can actually find it. It's this one, right? And it's connected directly to each one of the others.
Right, but that higher dimensional simplex is ordered, right?
Yes.
So if I'm going along the ordering, that means that the first one is connected to the second one. So they're all in this pathway that's 8 long.
Yes.
And then you also have connections between
That's
pairs
exactly
that are
right.
further.
That's exactly right. Yeah. I mean, it's all-to-all such that you can find a maximal length path, basically. Right? So if you have a maximal length, then you also have all the sum paths. Okay. Right. So that's where we are. So what you see here is basically the dimensions, the simple dimensions of the flood complexes that we generated. The blue one here is the actual reconstruction. This is the average of all 42, so it's pretty consistent. Here the shaded area is the error bar. And so it's pretty consistent. You get about 80 million 2 simplices, you get about 65 million 3 simplices, then it drops down. But it doesn't drop down to 5. What you see here in dimension 6, for instance, you still have 4100, and in dimension 7 you have 18. These are averages, okay? By comparison, the Algebraic one is the green one down here. It's pathetic, by comparison. The other two randomizations, including the Peter's rule, so the two biological randomizations are the ones that are here in yellow, and then the Peter's rule is the one just below it. So these are close to each other, so they're pretty good. I mean, this is the Peter's rule is pretty close to the biological randomizations, but it's not nearly as rich as the actual thing. Okay, So in other words, what you're seeing in the system, whether or not it's the actual brain, and nobody is claiming, including the scientists who built the system, nobody is claiming this is the actual brain of a rat, okay? But it is as close as you can get, and what you can see here is that it actually is very different from any other model, probability-based model that has been done so far. Okay, so, well, we're topologists, so when we build a complex, when we build topological space, we want to know its homology. Why not? Right? The problem is that when you do, when you want to compute homology, it's very nice if you want to compute homology of something which is, relatively small. But when you have something that has 8 million edges, right? It's pretty difficult, even if you have a computer scientist as clever and skillful as Pavel. So, in particular, as Pavel explained to me at some point, I insisted that we have to be able to do that. He explained to me that, well, look at the case where you have the two simplices and the three simplices in the complex. You have 80 million two simplices and 65 million three simplices. So imagine what the third differential is going to look like. It's a huge matrix. Even to just write it down is going to take too much space. And compute with it, forget it, difficult. So we're crushing in memory all the time. But you don't give up. You have a complex which is 7-dimensional. It's the highest dimension of a simplex we found. So what you can do is you can use what we call the N-core skeleton. So if you have a complex and you want to know its nth homology, and assuming that you have n simplices in the complex, you look at everything that goes above dimension n, all the simplices above dimension n and above. Then complement down, take all the faces, and the homology you're going to compute from dimension n and above is going to be the same homology as the homology of the original complex. So we did that. We took the 5-core skeleton of these complexes, and we were able to show that the top non-vanishing in all 42 columns, the top non-vanishing homology was in dimension 5. Okay, so they're all homotopical dimension 5, if you wish. We could see that they are connected. We were not able, however, to see any other homology. We are kind of trying to work on this now just to see what's going on. But we know that all of them have top homology in dimension 5. Okay, let's look at some results. So these tables here show, on one hand, the not so easy to compute fifth homology of all 5 rats. And the dots represent the actual rats. This is the Bedlewo 5, right? And on the left-hand side, they're very easy to compute. Once you have the complex, they're very easy to compute Euler characteristic. And I mean, okay, so this is, so you get some data here, so you get some numbers here. They're not very informing, or at least you could ask a very reasonable question: what's the point? I mean, why are you taking a brain? Let's say it's a brain. Why are you taking a brain and computing things like its fifth homology and its Euler characteristic? And it's a very sensible question. But scientists, they do things that we don't even consider doing. For instance, why would you take the fifth homology and the Euler characteristic and graph them against each other in a table, like in a graph, in a two-dimensional coordinate system, like this? Well, because sometimes you see something surprising. I mean, what you see here, basically, each one of the color blobs here is basically the kind of the center of mass of each of the rats, each of the individual rats. So here's this would be rat 3, and this one is thing number 2, and this one is number, I don't know which one is which, but it doesn't matter. They're all different colors. So you see that these 2 bits of information actually separate the 5 rats into 4 groups, where these are totally separated from each other. And these 2 seem to be brothers. Now we have no idea what the meaning of this is. This is entirely based on statistical biological data coming from the actual biological specimen. Why it expresses itself in this way, we have no idea. We don't have any idea what the meaning of it is. All we know is that it actually, it separates them. It's kind of interesting. Okay. Oh, wait. So we are applying these techniques to a digital reconstruction. We have to have, I mean, with all respect to blue-brain people, we have to have some sort of reality check, sanity check. So, well, we have a brain that we understand perfectly. We understand that brain, we understand as well as we understand the blue brain reconstruction. We know all the connections. So we do the same calculations for c-elegans. And here are the results. So what you have here is basically the complex. You see the complex looks different. It doesn't have this spike and then coming down. And this is the homology. This brown here is the homology. And we compare it to a Algebraic random graph with the same probabilities. And you can see the graph and the homology. Again, looks pathetic by comparison. The point is that we have a very well-defined, very strong difference between the Algebraic and the actual biological specimen, the c-elegans. Okay, so the rat and the c-elegans have non-trivial brains, topologically speaking. But if you're a biologist, that's not going to be very appealing to you. One question you'll ask, is it reasonable for the brain to know algebraic topology? And this is a good question. I mean, our brains here collectively, we know algebraic topology, but this takes a lot of training. Okay? The actual tissue, does that know algebraic topology? Well, you could ask, do the simplices have any biological meaning? For instance, do neurons in the simplex fire together? Is the activity in neurons better correlated? This is the same, this is another version of the same question. Is the activity in neurons in a simplex better correlated compared to other constellations of neurons? I mean, you can think of other sort of motifs, if you wish, other small graphs of neurons. Is it better for a simplex than otherwise? Also, are directed clicks more or less prevalent from arbitrary undirected clicks? Okay, so by click I mean this simplest I was talking about. I didn't actually define the word click before, but you understand what I mean. So, is there sort of preferential treatment in the system to directed clicks? What role is played by directionality in the simplex? Is there a notable difference between the functionality in a directed click compared to an undirected click? Well, I'm not going to tell you much about this because Catherine is going to tell you about it on Tuesday. Okay? So, turns out that the answer to these questions is pretty well understood, and but you're going to hear about that from Catherine. Okay, so now I want to talk a little bit about activity. I told you this is not just a structural graph. We actually have a system that can be stimulated. And I'm going to talk very little about this. I'm just going to describe the system of how we construct this sort of time series of matrices, of connectivity matrices corresponding to an active system. And then you'll hear more from Catherine about what we actually discovered. So what's the system? We're looking at the structural matrix. Okay, so let's basically stimulate the microcircuit in time intervals of 50 milliseconds, and you look at the reaction in a whole second. So you have 200 time bins where you have the reaction. If A is a structural connectivity matrix, then you can construct the successful transmission matrices where AK, for K between 0 and 199, AK will be 1 if and only if the following three conditions are satisfied. 1, neuron I is actually connected to neuron J. Okay, they all are. I didn't say that. The neurons in that system are all numbered. Each one of them has a name, which is just a number between 0 and 31,000. So in AKIJ, the IJ entry will be 1 if 1 that actually has a structural connection. So neuron I is connected to neuron J. If neuron I fired in time bin K, K is the upper index here, if neuron I fired in time bin K, and if neuron J fired within 7.5 milliseconds after neuron I did. And this number here is optimized by experimentation, and if these three conditions are satisfied, we put a 1 there under the hypothesis that there is a causal connection between the firing of neuron J and the firing of neuron I in about 7.5 milliseconds before. Okay? So we get a bunch of matrices, 200 matrices. And so here's what we did. I mean, this is like one of the initial experiments. Catherine is going to tell you about much more exciting ones. So this is called what we call a point versus circle experiment. This is stimulating the system in the form of a point and the form of a circle. What do I mean by that? You basically stimulate, you take the cross-section of the column, and you stimulate neurons which are spread around like in a circle, in here, and like in a point in here. Okay, so this is where the stimulation actually starts. You don't see anything here. This is the system starting to react. By the time you get to 15 to 20 milliseconds, it's very difficult to distinguish them, and then of course later on it's very difficult to distinguish. So these look like two different ways of deflecting the whiskers of the rat. I mean, this, if you want an analogy of what is actually happening in a rat's brain in this sort of an experiment, this is what's happening. Now, a very common way for neuroscientists to look at the reaction of a system is to look at the average firing rate of the neurons. So the neurons are firing at a certain rate, and looking at the rate in which they are firing is a common way of checking the reaction. And as you can see here, these are averages. As you can see here, the circle and the point create almost the same reaction, which is a good thing, because the idea was that we're going to try and compare the reactions, but we have to make sure that firing rate cannot actually properly distinguish them. So they try to make sure that the firing rate in both cases will be the same. Okay? And as you can see, they are indeed the same, more or less. Okay, so then we looked at our matrices. And what you see in these graphs is basically things like the one-dimensional simplices, two-dimensional simplices, the Betty 0, Betty 1, Betty 2, and Euler characteristic. And you start seeing differences. This is the time axis, and then you see the numbers of of all these parameters, all these topological parameters. And you can see that, for instance, for the point in the circle, if you look at the two-dimensional simplices, there is a notable distinction between them. You also see the Betty 2. There's a notable distinction. The Euler characteristic actually is a very good separator. These are, of course, averages. You have to be very careful to draw conclusions from that, because if you look at each individual experiment, and we ran dozens of them, each individual experiment, they are very difficult to compare to each other. On average, they behave differently, but it doesn't mean that you actually identify the signal. All it really means is that you have, on average, different behavior, which is kind of encouraging, but it's not an amazing thing. The amazing thing comes later, not by me, by Catherine. Right. So here's another, another sort of way of looking at things. So what you see here is graphing, in the previous example, graphing the Betty 1 versus the Betty 2. So this is 0 to 5 milliseconds after stimulus. This is 50 to 55 milliseconds after stimulus. And you can see that the square ones are actually the circle, and the dotty ones are the dots. And you can see that there's different reaction. There's actually a very good distinction between the two cases. Okay. But again, you have to be careful, because you see that they are spread out. These are pretty clustered, but these are pretty spread out. So it's not exactly the same reaction. Each one of these is an individual experiment. Okay, each one of the dots here is an individual experiment. So it's not really identifying them, just telling you they're not doing the same thing. Okay. Right. We then ran some statistical tests. We did the Gaussian base classifier. And we observed that, like we were hoping and kind of knew already, that the topological metrics actually does a better job in identifying, in separating the two signals than any others, particularly the Betty 2, and the Euler characteristic are doing a very good job. As you can see in here and in here, almost 100% of classification, of chord classification. Okay, and so and this is more recent, and this is 3 different experiments, again run many times, of different types of stimulations. You can see a little bit here how you're stimulating the neurons in certain groupings, and you see the reactions. So you see the reactions do look a little different. And then when you apply, here what you see here is the Betty 1 against the simplices of dimension larger than 3. And you see that there are different reactions. Okay, to these three different groupings, you have three different reactions, which is kind of interesting. Again, it's not a classifier, but it's something that shows you that there is a consistency in the way the system reacts to a given stimulus. Right, and more on functionality in Catherine's lecture on Tuesday, I think, or whenever it is. All right, so, yeah, so when you do apply topology and you come from a background of very pure topology, like I am, then you got curious about various things. So for instance, when you come up with this idea of directed flag complex, you could ask the question, what happens if you take just a simplex and you look at all its one simplices directed both ways? Okay, so you take a graph, which is basically a complete directed graph. So any pair of vertices is connected reciprocally. All right. And so let's call Gn the one that has n plus 1 vertices in made like this. Let Xn be the associated directed flag complex. And you can ask, well, what is that? What does it look like? And it turns out. So when I was asked this question by Oli Kramer about a year ago in Lausanne, I didn't know the answer, but I had Pavel's code. So I created a matrix that does that exactly. It's basically the matrix that has all ones and only zeros on the diagonal. And ran it through Pavel's code. And very quickly I started getting numbers. And the numbers were telling me that these things have homology only in the relevant dimension. And these funny numbers, well, you put them in Google and it tells you that these are the number of derangements of k points, or k plus 1, or n plus 1 points, where derangement means fixed point free permutations. Okay? So then you go and start thinking about this, and it turns out that it's not very difficult to prove that is indeed the case always. Okay, so the proposition is that if you have one of these complexes Xn, then the homotopy type is that of d n plus 1 n spheres, where dk is the number of derangements on n plus 1 points. Okay? I mean, I don't know a geometric proof of that, but you can prove it very easily in Euler characteristic, which you do know because you know exactly what the simplices are. So, for example, this little guy here, which is 4 points, all connected reciprocally. If you look at that, that gives you, if you take the directed flag complex associated with it, then it gives you space of the homotopy type of 9 3-spheres, a wedge of 9 3-spheres, and that has an action of sigma 4, which is kind of interesting because you know, I don't know of many spaces of the homotopy type of a wedge of 9 3-spheres with an action of sigma 4. It's in fact pretty difficult to imagine one of these, but here it is. So that complex, it turns out, I was wondering about this complex for a while. It turns out that people have been looking at this complex for quite a while now. It's called the complex of injective words. There are various equivalent definitions. I never saw in the literature anything like what I just presented, but if you read, if you finally find those objects in the literature, they are equivalent. It's clear that they are equivalent, and they're being used currently in the study of homological stability for groups, particularly for the symmetry group. You can take a directed graph and you can, an undirected graph, and then take the doubled. So for every edge, you double it. You take the two edges going both directions. And you can ask, well, what do you get when you take the directed flag complex of that? Nice question. Not applicable to neuroscience, because if you take neurons and connect them both ways every time, you're going to get a really problematic brain. Really problematic. So that doesn't happen, but theoretically it's interesting. And it turns out that if you do the same thing to a simplicial complex, so when we replace every n simplex by one of these xn's, then this is something that Oscar Wendell Williams and Natalie Levi actually looked at. And they have a theorem, which is a very nice theorem, which tells you that if you know the connectivity of the space, the original complex X, you can express the connectivity of that new complex, let's call it dx, in terms of the connectivity of X. Under some conditions, it's not for every complex, at least not in their paper, but under some conditions you can actually say something about the connectivity of that construction. And that can be used to do various things if you wish. So this is sort of a way of going back from applied topology, like really applied topology, to neuroscience, to a totally theoretical question, which is inspired by the applications. So K. Turner suggested the term applied topology. So there you go. Okay, what's next? So this is all I'm going to tell you about the Blue Brain project. Now, I want to tell you next about something that Catherine has, Pavel Notko and I are doing. And it has to do with highways. I was looking very hard in Google for a good picture of highways which look very impressive, and I found this one. I thought it's wonderful. So let's see what this is all about. So let's say that you have an order simplicial complex, right? You want to consider ways. So order simplicial complex for us, the motivation that we have is coming from neuroscience. Neuroscience is basically the study of systems of neurons connected in a directed way, and there's a flow of information which determines everything the brain does. So we want to consider ways of extracting flow and higher structure from such order simplicial complexes. So we define two notions which are in some sense dual. So if you fix a positive integer d, less or equal to the dimension of n, we call it the upper d highway graph associated to x, is the graph whose vertices are the d simplices. The vertices of the d simplices, I'm going to show you a picture in a moment so it will become clear. And the edges, there's a directed edge from sigma to tau if sigma and tau are the first and last phase in a directed d plus 1 simplex. Okay, so if you have a triangle, right, and the order goes like this, 1, 2, 3, then you'll have a directed one highway. We're going to have a one highway from the 1, 2 to the 2, 3. Okay, but I'm going to show you a better picture. Then, similarly, the lower d highway graph associated to x is the graph LDX, whose vertices are again the d simplices, and you have a directed edge from alpha to beta if the last codimension 1 phase of alpha coincides with the first codimension 1 phase of beta. Okay, so if they intersect on a codimension 1 simplex, which is the last of the first and the first of the last. Let's look at some pictures. Here's a picture, here's your graph, right? Here's a graph, a directed graph. And so here you can see how the upper highway graphs with the 1 simplices are the vertices, and you have an edge from 0, 1 to 1, 2 because you have, here's 0, 1, and here is 1, 2. So you have a directed edge from 0, 1 to 1, 2. And similarly for all the rest of them. The lower two highway graph, the vertices are the 2 simplices, and the edges are intersections of, for instance, 0, 1, 2, so here's 0, 1, 2, intersects with 1, 2, 5 on the last of 0, 1, 2 and the first of 1, 2, 5. Okay? Right. So, why would we think about it? Well, as I said, we want to extract higher structures. We want to extract information about flow. In directed flood complexes, more generally in any order simplicial complex. We could ask questions like, are highways particularly efficient information pathways in a neural system? This is something we can actually check. We can actually, either in an artificial neural system or with some luck, if we manage to program this in the Blue Brain reconstruction. Can highways be thought as computational modules in some sense? Okay, so I mean, they are, I'm going to talk a little bit about this later, within neural systems. How prevalent are they? So we did some sample computations within the Blue Brain reconstruction, and what we found is that there are 3,602 upper 4 highway paths of length 2. So these are three nodes. Okay, so length is 3 nodes, and there are lots of them. And mind you, upper 4 highway graphs mean highway paths. Mean that you have 4 simplex. And then a 5 simplex, another 4 simplex, then another 5 simplex connected to it. Okay, so lots of these, and then as the number of nodes go up, of course the number of such pathways goes down. So 130 of length 3, 1 of length 4. If you go for, instead of upper 4 highway graphs, you go to some 2 highway graphs or 3 highway graphs, these numbers become much higher. Okay, but at least we know that they are there. Right, let's make some observations. So, what are these highways actually? I mean, we call them highways because it's kind of a nice name, because we want to think about it as information pathways, etc., etc. In fact, information pathways which are better than just neuron-to-neuron connection. So, well, one observation is that they are actually a very efficient approximation for a larger simplex. So, if you have an upper n highway of highway path of length 2, right, like this, okay, I can actually write it explicitly, then all you have to do is connect V0 to Vn plus 2 and you have an n plus 1 simplex, n plus 2 simplex, sorry. So it's kind of an approximation for an n plus 2 simplex. Here's another, more generally you can say that if you have an upper n highway of path of length k, then the underlying path is only k plus 1 edges short of being an n plus k simplex. Here's an example. You have three two simplices. So this is an upper one highway of length 3. If you add 3 more paths, you get a 4 simplex. Okay, I hope that you can see this in the back. But if you just add these 3 more edges compared to the 7 you already have, you get a 4 simplex out of these 3 two simplices. Right. Another observation is that highway graphs are very good in detecting different structures in the directionality of the graph that you have. So these 3 graphs here are the same. They're going to give you all of them are going to give you something which, if you take the directed flag complex, what they're going to give you is the two-sphere. Okay. So homotopy theoretically, all 3 realize exactly the same thing. However, if you look at their highway graphs, they're all different. Okay? So the highway graphs are a good way of detecting variation in the directionality, even the geometric realization of the flag complex actually fails to do so. Of course, they're not unique, okay? Because, for instance, if you look at the lower 2 highway graph here and here, it's the same. Although the upper one highway graphs tell the difference. All right? So you don't have a uniqueness kind of statement, but you do see variations. And of course, once you have this concept, last time I gave this lecture in Ausdorf Institute in Bonn, I was asked the question, well, why do you only take codimension 1? Of course, you don't have to do that, and that gives you a way of thinking about these highway graphs as having more meaning, if you wish. So, if you look at an order simplicial complex as before, the upper nk highway graph associated to x is a digraph whose vertices, so it's unkx, whose vertices are the n simplices as before, with a directed edge from sigma to tau, if sigma and tau are the first and last phase in a directed n plus k simplex. Okay, so in particular, u and x, the one I defined before, is just un1 of x. And similarly, the lower nk highway graph associated to x is the digraph, lnk, this is an error, lnk, whose vertices are the n simplices and the directed edge from alpha to beta if the last codimension k phase of alpha coincides with the first codimension k phase of beta. And using this, we can actually define some graph invariants. So, and we find 3 of them. So the robustness of a pair of n simplices in an order simplicial complex X, you have a pair sigma and tau, denoted by rho sigma tau, is the largest k for which there is an edge from sigma to tau in unkx. Okay, so that means, I mean, to have higher robustness means that these 2 simplices are connected through higher simplices. Okay, now we don't take into account the length of that path in this definition. So if you wish to take the length into account, you have another invariant, and that is the r distance. So if you have an r larger than 0 and smaller than sigma and tau, the r distance from sigma to tau, which we denote by dr sigma tau, is the length of the shortest path from sigma to tau in u and rx. Okay, so of course, if you have a path of robustness k, if your robustness is k, then for every r you will have an r path from sigma to tau. And so you can ask, what is the distance? What is the r distance? And that gives you the answer. And finally, the r flexibility of a pair which we denote by FLR sigma tau, is the number of paths from sigma to tau in U and R of X. So, in other words, in how many ways can you get from sigma to tau through that graph? Now, these numbers could give you an idea about flow. We have to test it. I mean, this is not something that we can just say, oh, this is what gives you an idea of what the flow is. You really have to do some tests. We have to do some tests in artificial neural networks. You have to do some tests in general, maybe in more general networks. And we have to definitely do tests in the blue-brain reconstruction. But you can ask the question, okay, but why, in these definitions, why are you using only upper highways? Why not, what's wrong with lower highways? Well, it turns out that upper and lower are strongly related to each other. Remember, I said the title of this part of the lecture was Highways and Line Graphs. So, what is a line digraph? Suppose you have a directed graph, a digraph. The line digraph associated to the graph G is the directed graph delta G with v hat, e hat, omega hat. Omega is the direction function, such that v hat is just the edges of G. E hat is the pairs in E cross E, E prime and E cross E, such that the target of E is the source of E prime. And then the orientation is given just by the inclusion into V hat cross V hat. Okay, so let's take some examples. So for instance, if you have in G just one vertex, then delta G is empty. Right? If you have a single path, well, then it's just one point. If you have a reciprocal connection, you get a reciprocal connection. If you have three vertices with arrows going either out from the center one or into the center one, you get two distinct points. Okay? Things become a little more interesting when you have some more complicated graphs, but for instance, in this case, you still have three distinct points in the associated line digraph. Similarly here, this guy here, when you actually have a flow, you see you have a flow from here to here, and this is depicted in this edge here. You have a flow from here to here, that's depicted in this edge here. Okay? But let's get a little bit more interesting. It turns out that the reason to study these things is because if you're looking at the D lower highway graph of an order simplicial complex, this is nothing but delta, the associated line digraph to UD minus 1 of X. And that is why you don't have to define these graph invariants in two different ways. It's enough to do it in one way. Furthermore, this is a very nice observation because it gives rise to another proposition. If an order simplicial complex, and then for any integer between 0 and the dimension of X, one has the following sequence of inclusion. So the upper n highways highway graph is a subgraph of the lower n highway graph, or rather isomorphic to a subgraph, which is isomorphic by this proposition to delta of un minus 1. So you get an inclusion of un in delta of un minus 1. Delta preserves inclusions, and therefore you have an inclusion of that in delta of ln minus 1, which is isomorphic to delta squared of un minus 1, etc., etc. So at the end of the day, unx can be regarded as a subgraph of delta n times, so n, the repetition of n times line digraph of just the one skeleton of x. So the one skeleton of x, as you expect, because the connectivity of your graph actually determines everything. That's what it's supposed to be doing. And in fact, what this is telling you is that the upper n highway graph is actually a subthing of just repeat n times the construction of the associated line digraph of the one skeleton, the directed one skeleton. These things have a totally different flavor and are not, at least in my opinion, not quite as interesting when you don't consider orientation. But you can define line graphs associated to graphs without orientation. They're just, in my opinion, less interesting. Right, let's look at some examples. Now we are looking at the same picture as before, without the realization here. Same picture as before, except now you can see that you have that how the lower two highway graph is actually the line digraph of the upper one highway graph. Okay, so for instance, if you're looking at the path that goes from 0, 1 to 1, 2 to 2, 5, here you see a path from 0, 1, 2 to 1, 2, 5. Okay, and so on. So if you actually look at this picture carefully enough, you'll be able to see that indeed in these examples, what you have is that for every one of these paths, you have a corresponding, for every path of length 2, you have a corresponding path of length 1 in the lower 2 highway graph. Another example, things become more interesting when you have reciprocal connections. So what we did here is we took two simplex with reciprocal connections, all reciprocal connections. In black you see the original simplex, and the associated line digraph is in red. Okay, so you get a pretty interesting structure. As you can see, when you have a reciprocal connection, you get a reciprocal connection as the associated line digraph. But these two are not reciprocal. I mean, they go both ways, but they're not reciprocal. So you get more interesting pictures once you have reciprocal connections. Why am I telling you that? Because in the brain, biological or reconstructed, there are lots of reciprocal connections. So it's very important to be able to analyze those, and this is one way in which you can see that, and well, you get more interesting pictures than just what we had before. This guy here. Okay, well, the graphs are actually pretty simple. Right? So these are much more interesting. Okay. So finally, we need to do some theory after all. We want to think, well, what are these line digraphs? We want to know what they look like. So, for example, if you're looking at a digraph associated to a corolla, right? That's easy to see. Then the corresponding line digraph is just a bipartite. Right? For every path of length 2, you have a path of length 1. Right? Every path of length 2, you have a path of length 1. These are connected, basically all three connected to all 2. So what you're going to get is a bipartite 3 to 2. All right? And indeed, so you can kind of, since you know that every directed graph is made out of corollas, this tells you that, it suggests actually, that every line digraph is going to be made out of bipartites. And there's a theorem from 1960 of Harari and Norman, which says that if you have a digraph, it is a line digraph if and only if there are two partitions of the vertex set into these joint subsets, such that the edge set is a union of bipartites. So that's an old classical theorem about a characterization of line digraphs. Another thing that this is not quite characterization, but another thing that you realize when you start thinking about these things is that if you have a digraph without self loops, by self loops I mean loops that start and end in the same vertex. If you have a graph like that, then the directed flag complex of the associated line digraph is one-dimensional. In other words, the associated line digraph will not have any directed 3 clicks. Okay? If X is an ordered simplicial complex, then for each D larger than 1, the directed flag complex of the upper D highway graph is also one-dimensional. So these things are really very very one-dimensional, which is kind of surprising. But anyway, that's the way it is. And this is not very difficult to prove. It's actually fairly straightforward. Once you see what you have to do, once you see how they are built, it's rather straightforward. But it took us quite a while to see it. We've been thinking about these things for quite a while, and it took us quite a while to see that this is actually the case. In fact, you can prove more than that. So if you have an ordered simplicial complex and k is less than n, which is less than or equal to the dimension of x, then if n is larger or equal than 2k, the directed flag complex of u and kx is one-dimensional. Okay. So this sort of graph that you are getting in this way by thinking about highways are special. They're not just any graph. They have some special features to them which might inspire you to think about it in different ways than just arbitrary graphs. Keeping in mind that they're actually built out of simplices. They're not just graphs. They're built out of simplices. Every node in them is a simplex. Every connection is either an intersection or going up along simplices. So it could have some sort of computational meaning if you're thinking about it as a part of our neural system. Right. So wait, let me go back. About 10 years ago, Catherine and I started thinking about something or other, to do with neuroscience, to do with the Blue Brain project. We just started doing that. And we found a characterization theorem which seemed to be brand new. Not the one by Harari and Norman, something different. So the title of what I do here is not entirely new characterization theorem, not because we discovered it 10 years ago, as I'll show you in a minute. So I want to present this theorem. It's kind of a cute theorem. So here's the definition. If you have a directed graph, you write V, for a vertex V in V, you write V is less than W if there is an edge from V to W in G. And then you define two relations, and I'm going to define them formally and then show you some pictures to demonstrate what these relations actually mean. So the overlying relation on V is the transitive closure of the relation V1, V2 are in the relation if there exists some W in V such that V is less than W and V1 is less than W and V2 is also less than W. Okay? The underlying relation on V is the dual thing, the transitive closure of the relation V1, V2 in the relation if there is a W such that W is less than V1 and W is less than V2. So, what does it mean? The overlying relation from of V1 and V2, V1 will be related to V2 by the overlying relation if there's a sequence like that of vertices which are related in this way. Okay? Take the transitive closure, I mean basically V1 is related to that guy is related to the next one, etc. And if you have a chain like that, you have V1 over related to V2, overlyingly related to V2. And similarly, the underlying relation is just the dual graph. V1 is related to V2 with tilde lower u if you have this sort of graph. And the theorem says the following. And as you see, this theorem was, well, it turns out this theorem was proven in 1964 by a guy named Ushen that published it in a journal that we never heard of. In fact, it's very difficult to find even if you look pretty hard. I don't know how Catherine found it, but she did. So this was in 1964. He proved a theorem which actually implies, it's not exactly the same formulation, but it implies ours, and we rediscovered it in 2006. So you have a directed graph G. It is a line graph if and only if for all V1 and V2 in V there's at most one E, one edge, such that the endpoints of E are V1 and V2. In other words, you're not allowing any doubles. And then, if V1 is over related to V2, and V1 is less than W, then V2 is less than W. And then, if V1 is under related to V2, then and W is less than V1, then W is less than V2. A nice consequence of this, of our proof of the theorem, is that if you have if every line graph is associated to some digraph, okay? So they are special, but every line graph is associated to some digraph. Namely, given a digraph G satisfying the above conditions, there is some digraph sigma G such that delta of sigma G is G. Okay, so we can I mean, it's not unique. This is important to point out. This is not a unique graph that gives rise to a given digraph, but there is always one. Okay? And then, so I want to end by asking some questions. So, back in the context of neuroscience, again, how are highways related to information flow? Are they related to information flow? Are the concepts of robustness, R distance, and R flexibility meaningful in this way? And then another interesting observation is the following. Back in 2006, we started working on this. We thought about the connectivity graph of the system, of the blue brain system, and then about sort of a dual graph, namely where the vertices will be not the the neurons, but the connections, the synapses, if you wish, kind of grouped together, because you can have more than one synapse connecting two neurons. But the connections are the vertices, and the edges are actually the neurons connecting them. And it turns out that what you get is, well, you get the associated line digraph. That's exactly what it is. All right, that's where this theorem comes from. That's that I just stated. The graph is far bigger, and the question is, does it make sense to consider these graphs, because they're now special of a special type. They're not just any graph, right? The line digraphs. Does it make sense to consider these graphs or any part of them as computational units? Okay. Does it have any computational meaning that you can associate with bits of this very large, gigantic, in fact, digraph? And, well, I already asked this question, can we think about highways as sort of computational units? That's something that, these are all questions that we are going to, I mean, this is a project which is still in progress, and these are all questions we are hoping to answer sometime in the near future. Right, so finally, I want to do some last minute propaganda for a conference coming up in Applied Topology in Sapporo in August. 8 to 12, it's going to be a wonderful conference. If you haven't booked your trip yet, then please do so quickly and come join us. It's going to be wonderful. And that's all I have to say.
Let's put some light on the picture.
Why not?
Any questions for Ran?
Yes.
So in that last rediscovered theorem, some of the statements look like saying that something was universal in some category, because there were like arrows. So you had some vertices, and you were saying, well, if there's some arrow this way, then there's some arrow that way. As you were talking about final objects, that
I'm not sure this can be interpreted in this way. I mean, maybe we never thought of this. I never thought about it this way. But I mean, you have to kind of, when you think about what the line digraph actually is, you have to try to see, you know, if you're given one, how can you go back? All right. And so basically, these conditions tell you that if you have that, then you can, you know, you can reconstruct a graph from which it comes. I don't know another way of thinking about it, which will, you know, be more categorical, if you wish.
Just to double check, at the very beginning you said, you talk about, well, you described one of your basic objects to work with, ordered abstract complexes.
Yes.
And for what I understood, in one of those things, you can have some set of vertices with some order, and the same set of vertices with another order?
Yes.
Okay, and those were two different simplexes.
Totally different. So if you think of a two simplex, for instance, right, two simplex has three vertices. But if you basically if you take all possible permutations on three letters, that's going to give you six different simplices based on the same three vertices. So, and if you realize that, then one of these statements there tells you that you get, in this case, you're going to get two spheres with an action of sigma 3.
Okay, yeah.
But they're difficult to imagine. I try to draw it. It's only two spheres, right? It shouldn't be that difficult. I try to draw it. It's not easy to imagine what it looks like.
Any further questions for Ran? So I think it's a good time to start our official part of the evening. Let's thank the speaker again.